And welcome to this year's online Soapbox Science Milton Keynes event. My name is Laura Crook from Rothamsted Research, and I'm delighted to be your host for this afternoon. Soapbox Science is an outreach platform promoting women in STEM, highlighting the fantastic work they're doing and bringing science to the streets. Following the success of our first event on Thursday, we now have our remaining 5 fantastic scientists ready to tell you about the amazing research they do. And don't forget, if you missed our first 6 speakers on Thursday or want to watch today's event again, then the links will be there to catch up with afterwards. The format of this afternoon will be that each speaker has 5 minutes to introduce themselves and talk to us about their research. We will then have 5 minutes available for questions. So if you have any questions about their work or their life as a scientist, then please make sure you type them into the comments or chat in YouTube and Facebook, and we will be able to ask the speakers during the question and answer session. Our first speaker of the afternoon is Dr. Esther Garcia Cela, a mycologist at the University of Hertfordshire.
I'm Esther and I'm a mycologist. What that means? Means that I study the ecology of the fungi, but not this fungi. I study this fungi. This is Penicillium roqueforti. Penicillium roqueforti, we use it to produce tasty food like a cheese. Maybe you have seen this before. This is the famous plate of Alexander Fleming. Alexander Fleming discovered the penicillin using what? Using this fungi. This is Penicillium notatum. Penicillium notatum produce some chemical compounds that release in the media and inhibit the growth of this bacteria. And then now we have here an apple that is contaminated with Penicillium expansum. Penicillium expansum grow in the apple producing patulin. As we discussed before, some secondary metabolites on fungi are really beneficial for us as the antibiotic. However, other kind of metabolites as mycotoxins are really poisonous and can produce damage in the consumer. Regarding to patulin, patulin is a public health concern because can produce damage in a lot of diverse biological organisms as bacteria, mammal cells, plant, and even humans. In fact, in human, after the consumption, we can suffer damage in the liver, kidney, and spleen. The European Commission has regulated the maximum amount of patulin in apple juice, but also in some solid apple product as apple compound or baby and young children foods. In particularly, in this case, we cannot have more than 10 micrograms per gram of a final product. And then the recommendation for the industry is remove all the apples that are contaminated, even this one that have like a tiny spot of mildew. But now, what is the consumer behavior? If you have this apple in your house, are you throw it away? Are you eating? Or are you cutting a piece? And if you are cutting a piece, how much are you cutting? Even that microbiology or toxin release in food is a really new research. I believe we have some preliminary result that tell us that if you have in an apple one lesion that is up to 3 centimeters, you should cut at least 1 centimeter of the surrounding area of the contaminated apple. Even in the case of marmalade of apple jam, we found patulin across of the whole jar when we divide it in different layers of 2 centimeters. For this reason, I believe that even migration of toxin in food is a really new research area. This is important because we need to have strong and reliable scientific data to create guidelines to support the consumer in the decision that they took before to consume this kind of product. But also, as I told you before, the recommendation for the companies is don't use any product even that they have like a small amount of a small presence of this fungi. And then if we are able to produce specific guidelines and then remove only this part that is contaminated and then save one, we can also reduce the food waste. And then now, thank you for listening. I hope that the next time that you see a moldy apple in your house, you're thinking twice before to eat it. Thank you.
Thank you, Esther, for your great talk. That was brilliant. Thank you. I'm now going to hand over to Jack to ask you some questions.
Hi there, Esther. That's a fascinating talk. We have a few questions coming in. So I'm really fascinated about where these actual fungi and mycotoxins, where do they come from?
Well, in most of the cases, they can come from the soil. This is the case of penicillium and also for the environment. Most of them are saprophyte and then they live on the soil, but also some are in the air, sorry, and they can be across of all the environment that is surrounding to us.
Great. Yeah, it's always interesting to know exactly where, how your food is getting contaminated. And you're talking about those great illustration of the apple, everyone's seen the sort of moldy apple sitting in their fruit bowl. So you're talking about cutting away 1 centimetre of that apple. So, is that distance just for apples, or does it matter what the food actually is and where the mold is?
The problem is this is a really new topic and then we don't have enough research. Most of the research that is have done only in apples, but is this we can find another product that are contaminated like lemons or pears or and so on. The really difficulty in regard of the toxin is like they are thermostables, and then even if we process the fruit, we are not destroying them, and then we try to avoid that they really go inside of the food processing.
Great, thank you. And I'm always trying to get my kids to eat things, so they see that sort of moldy cheese that you showed us earlier. What's the difference between you talk about these sorts of molds that are dangerous for us to eat, and yet we eat the moldy cheese. So can you explain maybe what the difference between those?
Yes, the initial difference of now that the species that we speak about this for cheese, sorry, that we are using Penicillium roqueforti. In the case of apple is Penicillium expansum, or for example in the antibiotic is Penicillium notatum. Initially we have different species and then we expecting that they produce different secondary metabolites. But because this is secondary metabolite, it's not something that the fungi really need to grow. Sometimes, even in the same species, we can find a strain that are able to produce or not. This is other of the kind of question that we need to reply. We need to discover why these strains are producing this metabolite that apparently they don't give them like any competitive advantage against other strains from the same species that doesn't produce toxin.
Okay, that's great. And we've got a question that's come over from Facebook actually. So what are the sorts of products that are most contaminated in our diet usually?
Well, related with mycotoxin, mainly the coffee. Coffee is one of the products, and also cereals, because fungi are able to grow in food products that have lower availability of water, and then these kind of products are more susceptible to be contaminated with fungi, sorry, that can produce mycotoxin, that other kind of product like cucumber that have a lot of water and then are more susceptible to be contaminated with bacteria.
Okay, that's great, thanks. And So, what's your advice then for, people at home? So what's the best way that we can store food to try and reduce the risk of, it going moldy?
Well, the first recommendation is try to don't buy too much food. If you are not really going to eat, don't store at home for longer because our house, normally the environment temperature is quite high. And then we create one environment nicely for the microorganisms to grow. And also the second advice is in the moment that we already spot that we have one piece of fruit or one slice of bread that is damaged, this means that we have a lot of contamination. And then you must not eat this one, but also the following slice. And in the case of the fruit, in the moment that we spot one fruit that is spoiled, because the spore contamination is quite fast, we need to remove this, but also clean very well our basket before to keep the left of the fruit that we have.
Okay, that's really great. Thanks very much for answering all of those questions. I don't think we've got time for another one now. Thanks for your brilliant talk, Esther.
Thank you very much.
Thank you. Yeah, thank you once again, Esther. If we don't get through all the questions submitted for the speakers in this session, then we'll be making sure they're all answered as a blog on our website, put out through our social media channels, or as a comment on the YouTube link. So please come back and check in a few days' time. Our second speaker today is Tolulope Elemo, a PhD researcher at the Water Science Institute at Cranfield University.
Thank you, Laura, for the introduction. Good afternoon again, everyone. My name is Tolu, and I'll be talking about slows and filters. Now, everyone uses water. I'm sure we've all used water in some form today. Water comes from in its natural sources like rivers, lakes, and rain. Is not always ideal for immediate use, and that's the main reason why we treat it. Now, one of the oldest methods of treating water requires filtering it with sand, and that's linked with the technology known as slow sand filtration. Now, a slow sand filter is basically a container filled with sand laying on a layer of gravel, and it ranges from different sizes, so it can be as big as a football pitch, like the ones used by large water companies, or it can be really small, like my resident in-house slow sand filter, made up of sand and gravel. That's simple. Slow sand filters are a very powerful and effective water purifying process. So it basically removes contaminants, both biological, physical, and chemical pollutants in water. So you have water that looks like this, is treated to look like this. It does this by forming a biofilm layer on the surface of the filter known as popularly called the Schmutzdeck, which is just German for dirty sand. This layer has some microorganisms and different species of microorganisms that affect the biological treatment of dirty water. Now, it's also environmentally friendly because it uses very little energy and it's just fed by gravity. So we all know that our population is growing, and it's estimated that in the next 30 years, that's by 2050, the demand of water would have increased by 35 to 45%. So your first solution would be, let's build new water treatment facilities. But for sustainability purposes, the question you should be asking is, can we make the existing facilities produce more? And that's the basis for the project on which I'm working on. We're looking at how we can get more out of slow sand filters by getting more water out of existing assets without having to build new filters. Now, one of the key parts of slow sand filters is the process of cleaning. It requires the filter to be drained down and the sand removed. And the sand will be washed. This process is a bit time consuming. And so what we're looking at is a faster way to clean the filters so that that period can be used to produce more water. Now, my project, what I do on a daily basis is looking at ways that this new cleaning method might affect the filters. Of course, we want to make it produce more water and make it faster, but we also don't want to compromise the benefits of slow sand filters. We also want to make sure the water quality is still effective and it's still as environmentally friendly as it is now. So in conclusion, just some key points to remember. We all use water. The population is growing more and more, and we're going to need more water. And so simple technologies are working and they're effective. And instead of thinking of building new ones, we need to be focusing on improving the ones we already have. And researchers like myself are looking at ways to improve technologies like slow sand filters for us to get more water out of it. Thank you.
Hi, Tolu. That was really fascinating stuff. I've got a load of questions for you here. We're wondering really where does the sand come from? It sounds like you need to have quite a lot of sand to run all these filters. Where do you get it from?
Yes, so the sand is basically from, it's sharp sand. So it's basically the sand used for building or sand in the garbage that is used for source and fill. It's just sand found locally, although some soils like clay soils are not really ideal that have a lot of silt. But any sandy soil would, any sandy like sand would be sufficient for a slow sand filter.
Wow. And how widely are these used across the globe?
So they're used in almost everywhere. Almost all continents use slow sand filters. So you have the very large scale slow sand filters in places like, for example, in London, about 80% of London's water supply is via slow sand filters. You have them in the Netherlands, United States, Japan, but you also have places like in Mexico where there's a lot of household slow sand filter being implemented. So like I said, it's can get from really very big to really small household sizes. So it's widely used.
Wow, fantastic. We've got a question here that's coming from YouTube, from Terrera. She asks, taking into consideration issues of sustainability, is the sand filtration technology a viable option for remote villages in developing countries?
Yes, it is. So in fact, there are quite a number of work done on implementing slow sand filters for small communities and remote places. It's a very simple technology. It doesn't require chemical additives. So it's really ideal for places that are remote and removed from um, urban areas. So they can easily produce high quality, clean water without any external help. And like I said, it uses little energy. So for smaller scale filters, it doesn't require any additional energy for, like, say, water pumping and things like that. It's gravity fed. So you can just structure it in a way that it falls through gravity and you don't need electricity to pump water.
Wow. And is this sort of thing just for drinking water, or could you use it for other things like swimming pools or heating systems?
Yes, because it's water that has been treated to drinking quality can also be used for any other potable uses.
Cool. I think we've got time for one last quick one. So you said it's more environmentally friendly because it doesn't need power to use the filter. But does that take into account the fact you have to replace the sand and when you're having to empty it? How does that account for the energy usage?
Yeah. So when the filter gets dirty, so that's when it reaches like a maximum head loss, and you have to remove the top layer of sand. That sand is washed and returned to the filter. So it's kind of fits into a whole circular economy kind of system, because the sand is not it's not removed and disposed. It's removed, washed, and reused in the filter.
I think we might have time for one more. We've got another one that's come in from YouTube, from Emma, asking how long is the process from dirty to clean water?
So it depends on the size of the filter, really. So for small filters, you can have from dirty to clean water. That's the time it takes to go through the filter. You can have from ranging as quickly as from 10 minutes to some that take about 5 hours. So it's mostly depending on the size of the filter. But if just to add to that, it's when the filter is first of all put in place, the filter has to run for some time to be able to develop that biological capacity to treat the microorganisms in the water. But aside, once it's that period is called the ripening period. But once the filter is ripe, then it's just effective to have water coming in and going out of it.
What do you see as the end point of your research? Do you hope that people will be using the things that you've discovered, but actually in real world filters?
Yes, so the aim is to have this new cleaning methodology being implemented in major water treatment works, just to hasten and to reduce the downtime, reduce the time it takes to clean the filters. So yeah.
So what does your day look like, your average day?
An average day when I have experiments looks like turning on filters, taking samples, doing some analysis myself, sending some away for analysis, inputting data on Excel spreadsheets.
So there's quite a lot of sort of repetitive work in that sort of day-to-day life of a scientist. But do you have any sort of really memorable moments? Like, what's been your favourite moment of your PhD research so far?
So one of the favorite moments is when so I currently doing the research with Thames Water. So when the prototype of the filter, it's the prototype of the cleaner that is being proposed was brought in. So yeah, that was quite exciting because it was something to break the dull and mundane sampling.
Fantastic. Well, good luck with the rest of your research.
Thank you.
Okay, that was brilliant. Thank you very much, Tolu, for your talk and your interesting props as well with that. Thank you for those questions too. So we will now move on to our next speaker of the afternoon, who is Luca Steel, a plant pathology PhD researcher at Rothamsted Research.
Thanks, Laura, and hi, everyone. Thanks very much for tuning in today. So I thought I'd start off by asking what these 4 foods have in common. So on this platter here, I have some bread, a breadstick, a cake, and some pasta. And one of the things these all have in common is that they're 4 of my favourite foods. But another thing that they have in common is they're all made using wheat. So wheat is a really important crop. It's important for the UK economically and also for most of Europe. But more importantly, it's a really important staple crop for billions of people worldwide. So it is in our bread and our pasta and our cakes. And we all depend on this day to day. As our world population increases, we're going to need more and more wheat from the same amount of land that we've always had. And a problem that we're facing is that just the same as you and I can get a cough or a cold or a disease, wheat and other plants can also get diseases. And that's a really big threat to food security. But it's not all doom and gloom. Luckily, science can do something about this. So my research focuses on one particular disease of wheat. It's called septoria leaf blotch. And septoria leaf blotch causes wheat leaves to go from being nice and healthy and green like this, to all kind of brown and blotchy and a bit just diseased looking like this. And what happens when wheat leaves look like this is you can see there's a lot less green area. So that means they can't do as much photosynthesis, they can't turn as much sunlight into sugar, and they can't grow as big. So we get less wheat. And actually, farmers who get this infection in their fields of wheat can see losses of up to 50% of their crop. So that's like, imagine it's payday and only half of your money turns up in your account, or you're in a restaurant and only half of your pizza comes out. That's probably how those farmers feel when they get this infection in their fields. So we need new ways to control this. Another problem of this disease is that obviously farmers are worried about it. They don't want it in their fields. Who would? So they tend to spray a lot of fungicide to protect against it. And this means that this one disease, septoria leaf blotch, is responsible for 70% of our fungicide use in the EU. And this is a problem because fungicide's expensive, but also the more fungicide we spray, the more likely it is that this disease will become resistant and we'll have no way of controlling it. So we need new ways to control it. And that's one thing my lab are looking for. So my PhD work focuses on the pathogen, the fungus that causes septoria leaf blotch. Now this fungus, it looks quite friendly, but it's actually the ultimate bad guy. It's called Zymo-Septoria tritici. It's a long name, so I just call it Zymo for short. And what Zymo does is it lands on the leaves of wheat and it's kind of like a master of disguise. So it produces a special chemical that acts a bit like a mask. And this mask hides the zymo from the wheat's immune system. So the wheat can't see it coming, so it can't fight it off, and so it gets infected. And what's interesting about this mask is if you take it away from the zymo, so if you stop the zymo from wearing this mask, it can't cause infection at all. So the mask is really key to the virulence of this disease. So I'm investigating this mask because zymo only wears this mask for very short periods of time. It doesn't wear it all the time. So I'm asking what triggers zymo to put its mask on? Why does it wear it sometimes and not other times? And the way that I'm doing this is we've got these lovely zymospores here, and we've edited them so that when zymo is wearing its mask, they glow green. And when zymo is not wearing its mask, they don't glow green. So what I can do is grow zymo under lots of different conditions to test when it is wearing its mask, when it's glowing green, and when it's not wearing its mask, when it's not glowing green. And we can test different conditions like different nutrients or different stages of growth. And hopefully, if we can identify some of those triggers, then we can stop those triggers from happening in the first place. So zymo won't be able to wear its mask. And if zymo doesn't have its disguise, then wheat will be able to see it, it will be able to detect it, and its immune system will be able to fight it off, just the same as your immune system might be able to fight off a cold. So that would leave hopefully more healthy wheat. We'd be able to make more of our staple foods with it and hopefully feed the population of the world. So that's a bit about my research. Thanks very much for listening, and I'll happily try and answer any questions you might have.
Thank you very much, Luca. That was fascinating, and I really liked your cuddly zymo with its mask. So I'm now going to pass over to Jack again, who will ask you some questions.
Hi there, that was really fascinating. I seem to get all the fungal talks here. It's really good. I've got a couple of questions. So that was brilliant prop that you used. So is zymo actually blue in real life, like your fluffy toy?
No, unfortunately, zymo is not blue in real life. I wish it was. It would make my life a lot more exciting. Zymo is actually so tiny that you wouldn't be able to see it just with your eye. When it's growing on wheat, you can't really see the zymo itself. Sometimes when it's reproduced, you can see tiny black little circles. So if anything, I'd say it was black. Under a microscope, yeah, it doesn't really have much colour, unfortunately. I wish it did.
Yeah, that's a shame. It would be good if we could work with lots of colourful things. So are there any other sorts of fungi that are sort of masters of disguise, a bit like this zymo thing? So are there any other types of fungus that have this sort of masked approach?
Yeah, that's a really good question. This is something that another research lab in the Netherlands are actually looking into. Yes, there are other fungi, some other plant pathogens actually that use a similar kind of mask with a very similar design. Something I've been looking into recently is do other fungi which aren't pathogens have a similar kind of chemical? And the answer is that a lot of them do. And that's really interesting. We're not really sure why. It could be to do with the fact that some fungi are kind of like friendly with plants and help them out, and they also need to be able to invade plants. But yeah, it's an active area of research, so good question.
No, it's fascinating. So we're getting quite a lot of questions in. It's obviously sparked a lot of interest for people. So we've got a question from Julia on Facebook. So what aspects of the wheat's sort of immune system do you hope will sort of kick in to respond to the fungi?
Yeah, good question. So the wheat's immune system is really interesting. It's something a lot of people don't even realise really exists. But wheat it has kind of like detectors that can detect signals from pathogens. So the main thing, if we can stop zymo from wearing its mask, then the immune kind of receptors will be able to see some chemicals that zymo produces. And then it will be kind of the very early stages of immunity. So the plant might be able to produce a chemical that can break down zymo, or something interesting plants do quite late on in infection is they tend to kill parts of themselves off to stop the fungus from spreading throughout them.
Oh wow, really?
Yeah, so it'd be like if you got an infection in your finger and you just chopped your finger off, stop it from spreading.
Yeah, that's a bit drastic, isn't it, really?
Mm.
Gosh, yeah, but obviously works. So we've got some more questions from YouTube actually this time. So is this problem with this particular leaf blotch more prevalent in some areas of the country than others? And that's from Jan.
Okay, good question. I actually don't know the answer to that within the UK. I've got some colleagues who will know the answer, so I will check in with them and let you know. But we do know it's a big problem across Europe. So it's particularly these European kind of temperate zones where Zymo likes to live. It likes really wet areas because the rain helps it to spread.
Okay, so not very good then for the UK with our high rainfall in some areas. We've got another question. I think we've got time for a few more, again from people on YouTube. So from Eric, it says, hi Luca, is there any reason for your research into this particular disease and not any other diseases that affect wheat?
Yeah, I really, so yeah, I did used to research another pathogen of wheat, another fungus that was quite similar. So that's kind of why I started kind of in this area on the fungal pathogens of wheat. This particular fungus is really interesting to research, I think, because we don't know much at all about it. We don't really know how it infects wheat as well as we do for other pathogens. And it's quite it's quite a lazy option. It's quite easy to research because unusually for a fungal pathogen, it has two forms of growth. So it can grow kind of like a yeast by budding and reproducing, and it can grow as kind of filaments like little hairs. So that means that we can it opens some quite interesting methods of research that we can use.
No, that's great. Well, good justification. And I guess the fact also that, you know, 70%, I think it was, of the fungicide use is due to this issue in the EU. So during your research, I'm quite curious about, you know, you were talking about testing all these different conditions under which you could see it, whether it had the mask on or not. So what have you come up with any results yet that show what those best conditions are, where you can see I forgot the name of it now, the blotch without the mask?
Yeah, it's early days, but yeah, we've got some kind of very early findings, so don't take this as fact yet. But something I have found is that the mask tends to kind of be switched on as the fungus just starts to germinate. So as it starts to stretch out its little like hair-like fingers to grow over the wheat leaf, it tends to be switched on then. So it could be something to do with that, could be something to do with some of the chemicals that are present on a wheat leaf, because what we've done is tested some different kind of soups that we've grown the fungus in. So we've grown them in a soup with some wheat leaves in and a soup with some other leaves in, and it grows best and it glows the most, the greenest in the wheat broths. So we think it's something to do with the wheat leaf chemicals.
Great, that's fascinating. So I don't think we've got time for any more questions, but thanks very much for that brilliant talk, Luca.
Thank you, everyone. Thanks.
Yes, thank you very much, Luca. Yes, some really good questions there. So our next speaker, Dr. Kate Hewins from the Cranfield Forensic Institute, unfortunately couldn't join us today, but we hope that she will record her talk and then we will make sure it is available for you to catch up with, because Kate was discussing a fascinating topic, so watch this space. So we will go on to our next speaker, Lois Damptey, a surface materials engineering PhD researcher at the Open University.
Thank you so much, Luca, for the introduction. So if you've been sitting down for a while watching Soapbox Science, you are just about to take a little exercise. So do you remember the song, I'm Walking on Sunshine? Oh, so we are going to go like this. I'm walking on sunshine. Oh, I'm walking on sunshine. Oh, I'm walking on sunshine. Oh, and it feels so good. Did you get that? So we are going to go the next time. You can sing the song and dance as well. So we are going, I'm walking on sunshine. Ah, I'm walking on sunshine. Ah, I'm walking on sunshine. Ah, and it feels so good. Okay, if you're wondering why I'm singing this song, whenever there is summer in the UK, I call some friends, we go to the park and have a good time to some good music. What do you do when the sun pops up? Do you go to the park to get some tan? Do you get some lollipop ice cream? Or do you play double dutch with your friends? In reality, I use sun to purify water. Did you know that 1.8 billion people do not have access to potable drinking water? Did you know also that every 2 minutes, a child under the age of 5 died out of waterborne related diseases? In some parts of developing countries, women and children have to suffer to walk long distances, about 1 to 3 kilometers, just in search of clean water for their families. So just by the rising of the sun, you're rest assured that you're saving a child from dying through a mechanism called photocatalysis. Photo, not your regular kind of selfie, but light, and catalysis, which means simply slowing or speeding up a chemical reaction. In this case, the chemical reaction is by breaking toxic compounds that are present in water that you cannot see with your mere eyes. Photocatalysis can be likened to plants undergoing photosynthesis. So in just 10 seconds in the chat box session, I want you to tell me what you remember about photosynthesis. And your clock starts now. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. I hope there are some interesting answers in the chat box session, and I hope we are all right. Simply put, photosynthesis is the process whereby plants make their own food using me as sunlight. In this case, plant has a material called chlorophyll that is able to absorb sunlight. So in this case, it makes the plant a photocatalytic material. So in the photocatalysis of water treatment, a photocatalytic material is very important. Over the years, titanium dioxide has been used as a photocatalytic material. This material is able to absorb sunlight to produce what we call electrons and holes. These electrons and holes are able to migrate onto the surface of the material, which is in presence and contact with the water body. And these electrons and holes are able to mineralize the toxic compounds that are present in this water body completely. So therefore, it's very essential to be able to choose the right material that is able to separate the electrons and holes without recombining with itself, therefore lowering the efficiency of this material. Although titanium oxide has been used as a viable material, it's only able to absorb 20% of the sunlight. So scientists like me are working tirelessly to tune the properties of this titanium oxide to be able to absorb the highest parts of the sunlight, which is 80%, which is called the visible light. So the impact of my work is to be able to fine-tune the properties of titanium oxides or create other materials that are used in conjunction with titanium oxide as composite materials that have higher efficiency to be able to absorb higher sunlight. Also, the wider impact of my work is providing access to potable drinking water, mainly by scaling up a production of what we call photochemical reactors in rural and remote areas in West Africa where sunlight is readily available. Thank you very much.
Thank you so much, Lois. That was great. We all needed a good sing-along for a Saturday afternoon. So thank you. And I love the sun as well. It's brilliant. So I'm going to pass this over to Helen, who should hopefully have some questions for you, Lois.
Okay.
Yeah, you really cheered up our afternoon there, Lois. Nice and bright and sunshiny.
Thank you.
So you you say that obviously sunshine is a really great tool for this sort of thing. But we know that sunshine can also be quite damaging to materials. So does this cause any problems when you're designing sunlight-based systems?
Okay, so mostly what we do in the lab is to simulate the sunlight processes. So this material are able to just tune their properties to sunlight without degrading them or without releasing their toxic metals into the water. So with that, in the process of our production, we are able to check the changing variable measures like the pH and other things, which obviously if a material degrades, then it's able to release some toxic compounds into the water, which we can test for. So in the course of every lab work, we are able to check that along the way.
Are there any substances or pollutants in the water that it's harder for sunlight to act on?
Yes. So basically, most of material properties are able to break down organic compounds. Okay. So ideally, one of the things that this material does is that they are researching on its antibacterial agents and other sources and its absorption methods. So sometimes they are not able to do certain parts of the water, like take away anti-fouling, the smell and all other things. But in tuning the properties or creating nanocomposites of this material by joining them with other things, they are able to take care of the other pollutants or any other thing present in the water.
Wow, fascinating. So you said currently the materials that are being used in current systems can convert 20% of the sunlight.
Yeah.
What do you hope we'll be able to do thanks to your research?
Yeah, so my research is looking at the fact that this material, there are new materials that are being formed from titanium oxide, that titanium carbides. What they do is that their surface properties can be tuned. Okay, so when you tune the surface property, it's able to absorb the highest part of the electron. And that's one thing about this material that makes it versatile. Yeah, so it's able to absorb the highest part of the electromagnetic spectrum, which is the visible light, which is 80% of the total sunlight.
Wow, so it would be much more efficient. And
yeah,
much quicker. In your promo video, we saw that you were using dye compounds, pollutants.
Yeah.
Do you do that just as an analog, or are they actually similar to pollutants you find?
Yeah, so this project is looking at areas in Ghana where there are textiles industries. So one of the things is that creating a model compound. So we are trying to test on them using dyes that are from textile companies that are being led into our water streams, which are main water streams for people that feed on them. Also, there's a pilot project in India that also uses much wider textile industries. So those are model compounds that this material has proven to be able to break down. So other properties of the material is coming up as we proceed in the research.
Cool. So we've got a question here from YouTube, from Eric, who wants to know, are there other applications for this research apart from water treatment?
So this material has been proving anything that is photocatalytic viable. So even in our solar systems, solar panels, they are used as base material because they are able to absorb the sunlight or anything that's coming up with solar efficiency projects, even touch lights and all that. They have a component of this material because it is able to absorb the sunlight.
So can you use other sources of light or energy in the same way then?
So we basically use just the sunlight. So we have a solar simulator system in the laboratory that has the intensity as the sunlight that it's in the open. Then we try it. Sometimes we take our test equipment outside and we use the normal sunlight that's available, and you can get as much efficiency as the solar simulator in the lab.
And I think we've got time for one last question. We heard earlier about sand filtration for water. Which is better, sunlight or sand?
Well, everyone complements each other. Mostly, I think, using things that are readily available to us. So with Tolu, she's using sand, which is readily available to us. I'm using sunlight, which is also readily available in that part of the world. So basically combining efforts to use things that are readily available to us to produce maximum efficiency to the people.
That's really great. Thank you, Lois.
You're welcome.
Yes, thank you, Lois. Yeah, that was brilliant. Thank you very much for those questions and for answering those. A reminder that any of the questions not answered today will be made available in the coming days afterwards, so please do come back and check the links to get even more information on our fascinating topics that have been discussed today. So our final speaker of today's session is Kate Hand, an urban ecology PhD researcher at the Open University.
Hi everyone, I'm Kate, and as Laura said, I'm an urban ecologist. Everyone see me. So I just want to explain what urban ecology is. So an ecologist is someone who studies the plants and animals and how they interact with their environment. And urban ecologist is just someone who studies that for urban areas, so our towns and our cities. Now, you might think our towns and cities are a bit of a strange place for an ecologist to work. Wouldn't it be better to be somewhere where in the wilderness, where there's like wildlife and proper nature? Well, our cities actually have really interesting, diverse and rich arrays of wildlife, which are constantly evolving as cities grow. So they're really dynamic and interesting places to study. But what I think is most important about urban nature is that the majority of the world's population now lives in urban areas. So the nature we see as we go around our daily lives, as we go on our commute, we walk around our neighborhood, we go to school, that's the nature that's going to be influencing our daily life. And fortunately, nature has the opportunity to do us a lot of good things. And in particular, urban trees are a big bit of urban nature, and they do lots of good things for us. So I want to just talk about some of the good things that urban trees do for us, and some of them you might be familiar with. So if we think back to lockdown, maybe when you were stuck inside your house, you really appreciated the fact that there is a tree across the street from you that you could look at. And perhaps you really appreciated the fact that that tree blocked the view of that neighbor you don't like across from you. Or perhaps you really appreciate the fact that that tree blocks some noise from a busy road across the street from you. Or perhaps you really appreciate the fact that you could look out from your window, see the trees, see its foliage changing with the seasons, or maybe see some birds come and visit it and listen to their song as they look for bugs in the leaves and things. So these all might sound like quite small things, but they all add up to improve our daily life. They help to lower our stress levels and improve our mental well-being. And that's really important, especially at times like this. Urban trees also do a lot of things that maybe you can't see with your eye. So urban trees help us to improve urban air quality. So urban areas have a bit of an issue with poor air quality, which means there's high concentrations of air pollutants in the air. And these pollutants can move through the air. And if we breathe in them in for a long period of time with lots of high concentrations, we can actually get sick. But what urban trees can do is that they can intercept these pollutants before we breathe them in. And this prevents us from breathing those in, and so it helps improve our health. The other thing urban trees can do is they can help us in the fight against climate change. So climate change is caused in part by carbon dioxide, or CO2. Now, as trees grow, they suck in that carbon dioxide and they split apart the carbon and the oxygen. They let the oxygen go up into the air, which is a good thing for us to breathe. And they store that carbon in their woody mass, so that's in their branches and the trunk and things. And every year, as the tree grows, it sucks in more and more and more carbon. So it gets bigger and bigger and bigger, and it provides more of this carbon storage service, locking that carbon up so it's not heating up the earth. So big trees tend to provide much more carbon storage than small trees, and this is typical for lots of other benefits. So for instance, a tree with much more leaf area, a bigger tree is going to intercept more air pollutants than a small tree. So that's going to be better for improving air quality. So this is true of lots of different benefits. And we can actually use tree size to predict the benefits that trees are providing us with. So this is what I do a lot in my research. And what I'm particularly interested in is how size of a tree is going to change over time and how that's going to change the benefits that that tree provides. So I'm interested in tree growth. How long is it going to take a little tree like this to get to this size, and how is its benefits going to change over time? And I'm interested in this because if we know how trees are growing, if we know what benefits they're providing us, we can think a little bit more about how we're going to manage our urban trees so that they can provide the most benefits for us. And there's lots of different things that could affect tree growth. So it could depend on the species of tree, it could depend where that tree is planted, or it could depend on how we manage that tree. So there's lots of different things that could affect the growth rate and therefore affect the benefits that that tree provides. So to find out more about urban tree growth, I've set up an experiment in Milton Keynes. To measure tree growth, we use a pretty simple bit of kit, but its fancy name is a dendrometer band. So it's essentially, if you imagine this is a bit of a tree, the trunk of a tree, the dendrometer band is essentially a bit of metal tape attached with a spring. The spring allows the band to expand as the tree grows. And we just watch this little arrow marker, and we can, as the tree grows, we can see how it moves along the tape, and we can track its growth. So we just have to go back to the same tree multiple times to measure its growth. And the more we go back and measure the same tree, the more data we can get about how it's growing and what's affecting its growth rate. And this is actually where you can help out. If you're around in Milton Keynes, you can help track the growth rate of trees. If you come across a tree with a band like this, there'll be a tag on the tree explaining what you can do to measure it. You don't need any specialist equipment, or just yourself. And the more data we collect on trees, the better we can find out about what's affecting their growth. This is actually the first time we're collecting tree growth in this way in the UK. So it's a really interesting new kind of study. And we're really excited because if it works, we could maybe expand it and collect much more data about urban trees, and we can look at what's affecting their growth. And we can ultimately, we can use that information to, we can look at that information to help to plan and manage our urban areas so that they're more sustainable, greener, and healthier places for us to live. Thanks.
Thank you, Kate. That was really interesting. I love the props, and I will definitely be looking out for your trees next time I'm in Milton Keynes. So I think I'll pass you over to Jack for some more questions.
Hi there, Kate. That's really fascinating talk. I hope everyone looks at trees differently in the cities and towns that they live in now. So I have a few questions. There's a couple that have come in already. So someone on Facebook. So what are the best type of trees to plant to capture pollution and also carbon?
Oh, well, that's a couple of different things. Really interesting question. So in terms of capturing air pollution, it's the trees that are really leafy that are going to capture air pollution. And actually, I think a lot of studies have shown that hedges are very good to plant because they capture pollution that comes from the emissions of cars. They can capture it when it's close to the ground. But also little things like tree leaves that are hairy actually catch more air pollution than kind of shiny or waxy leaves. So there's lots of different things to look at, and there is information out there, so I can send it out in the comments and link you to some nice resources at that point that tell you what trees are best for that.
That's great. So get planting hedges, and lots of hairy things are good then. So,
yes.
Yeah.
and then for carbon storage, trees that grow quickly are best for that. And just letting them live for a long time so that they can store that carbon for a long time is the best way to get to do that.
Yeah, so there's kind of like a related question to that. So, we've only got limited space in urban areas. So you're saying that big trees are best for all those things. So, how is there enough space for really big trees in urban areas?
Yeah, it's a really important question. And of course, yeah, big trees do provide the most benefit. But our urban areas are often crowded, and often you can't have them. So I think it's maybe thinking a little bit more about how we can design around our urban trees. If we're thinking about a new housing development, planning space for a big tree, or thinking about we're going to save this tree so it can grow into a big tree and it can provide for benefits. So we're not stuck with all these little tiny trees that are all just a little small and they're not doing too much. So yeah, it's a bit of a kind of, we need to work together with different kinds of urban planners to integrate trees much more so that we can get the best kind of urban areas that we want.
We've got a couple more questions coming through, so sorry, I'm keeping track of those over there. One from YouTube. So it's not about urban trees, but we're talking about, I guess this has been in the news recently, about cutting down ancient woodland. So how long do you think it would take to compensate and get the benefits back from that?
Well, ancient woodlands are really, they're not my area of expertise, but they are hugely unique environments. And the thing about cutting down an ancient tree is you can't just get that nature back. It's huge amounts of biodiversity that are associated with an ancient tree. An ancient woodland in particular have this whole kind of ecosystem that's built up in the soil over hundreds and hundreds of years. So you can't just replant a tree, wait for it to grow for 100 years. It's in no way the same, and that's why this environment is so valuable. And in many ways, it's just irreplaceable.
Yeah, it's really important. I'm glad you mentioned soil there. I'm a soil scientist, so I always have to put that word in there. We've got another question from Luca on Facebook. So does it matter whereabouts then you talked about it a little bit in your talk, where we plant trees in urban areas? So, are some places better than others? So where can we best position them for those maximum benefits?
Yeah, absolutely. So this is what a lot of research is looking into, is where best to plant, and then different species are better for providing different benefits. So it all kind of ties into what kind of benefit you're looking at. If you've got high area with high concentrations of air pollution, then you're looking at maybe planting a certain type of tree and planting it in a certain way, such as in the hedges, that you can collect that air pollution. Or maybe you want to encourage wildlife, so maybe you're looking at trees that provide fruit or flowers that's going to invite wildlife. So it is tying up the different characteristics of different trees, and there are lots of different areas that you can put them in. So it is quite an interesting way to kind of arrange the pieces on your urban planning map and put them all in to kind of get that best outcome.
Make space for trees, definitely.
Exactly, yes.
So I'm really curious about that band that you had. So you've got this thing that's now measuring. So, you know, it's really exciting, like using the general public, if you like, to take some, you know, scientific measurements on your behalf almost. So, you know, what inspired you then to sort of involve the public in your research?
Well, I think in particular with urban trees, it is really important to involve the public because, you know, the trees do play a big part in kind of, you know, our urban areas, and helping people to understand the benefits that they might not know about that trees are quietly doing away in the background. So part of it is just engaging people with urban trees and helping them to understand the benefits they provide. But also, I mean, hopefully a lot of people are really interested in urban trees and do want them to do well. So this way they can get involved and help towards a scientific progress. And hopefully it'll be something interesting that they enjoy and take part in.
I think it's always really interesting then to have a project that, you know, like you say, the general public are interested in the outputs as well, and it's directly relevant to them. I live in London, so, you know, I can appreciate what the trees are doing for me. That's really good. Thanks very much. Unfortunately, we don't have any more time for questions, so thanks very much.
So much.
Thanks.
And thank you very much for all of our questions and all of our speakers. And so that concludes our session for this afternoon and our online Soapbox Science events for 2020. Don't forget that the links for YouTube and Facebook from both events will be available for if you missed our first session on Thursday or you'd just like to watch them both again. We will also make sure that answers to all the questions that have been asked by our audiences in both sessions will be available as soon as possible. I would like to thank our speakers for their amazing presentations this afternoon. They have been fascinating with some brilliant props and inspiring science. I'd also like to thank everyone who joined our live streams today and for submitting your excellent questions to our scientists. I'd like to say a huge thank you to my fellow organising committee members for all the hard work they have put in to make these events happen, including behind the scenes now as I speak. And a special thank you to Katrina, Sam and AJ from the Open University for their technical support and expertise, allowing us to broadcast live for both our events today and Thursday. So we hope you enjoy the rest of your weekend, and we look forward to welcoming you to another Soapbox Science Milton Keynes event in the future.